So I'm going to be talking about our manifold embedding algorithm, Rastermap. So about extracting structure from high dimensional neural data. And so this is really finding kind of patterns of activity that you might not have been able to see without an algorithm like this. And briefly at the beginning, I'm going to talk a little bit about how neural activity, that we found that neural activity is high dimensional, because I think it's an important point to make when you're building models, particularly of visual cortex, that you keep in mind kind of that there are lots of patterns of activity that the neural space explores. And that using the high dimensional activity, the structure of that high dimensional activity helped us to create Rastermap. All right, so this first question is kind of what is the dimensionality of neural activity? And so you basically have these images that your retina sees, and these images get sent to your thalamus, and then they get sent to your visual cortex. And there's around 50 times more neurons in visual cortex than in thalamus, in mice and humans. And so you have this question of what is this expansion used for? And so if, for instance, every neuron in visual cortex, there's like 50 of them that all do the same thing as one thalamic neuron, then they would kind of be this sort of redundant low dimensional representation. But you could, for instance, imagine that each neuron in visual cortex is encoding different features that it extracts from this thalamic representation. And that would create kind of a high dimensional representation that might be useful, for instance, for telling you if it's a tiger or not. And so those kinds of high dimensional transformations are the sorts of things that happen in deep neural networks, for instance. And so basically, we kind of have these two hypotheses: is it low dimensional or high dimensional? And so low dimensional representations, for instance, would be if we have a single one-dimensional stimulus, and we have these tuning curves like this that are quite wide. You'd have a low dimensional representation of this one-dimensional stimulus space. So you'd have two significant eigenvalues, for instance, if we're computing dimensionality this way, and then you kind of have this smooth representation of this stimulus space. You could alternatively have a high dimensional code. And so here's kind of a high dimensional code, the sort of efficient code that people have thought of in the past, where every single neuron is coding for a different stimulus along this one-dimensional stimulus dimension. So you have these little spikes. And then each of these neurons has equal variance. So you have an eigenspectrum that ends up being flat. And then if you try to move through this one-dimensional space, it ends up being this kind of spiky ball, because you go from neuron to neuron, and there's a completely different neuron encoding every single position. So it's not a smooth representation. And so a lot of papers in the past have suggested that neural activity is low dimensional. And I've kind of made a summary of the papers that were, for instance, in this review paper from Gao and Ganguly. But there's an important point that this review paper makes about the dimensionality of these recordings that kind of is, in this case, around 10 or less, and also this paper here, that actually your dimensionality is limited by the number of neurons that you've recorded, and also the number of stimuli, or like the complexity of the task that you're using. And so basically in our study, we tried to kind of no longer restrict both neurons and stimuli. We recorded many neurons, and we showed around 3,000 stimuli twice to the mice in our recordings. And so hopefully we're not limiting our estimation of the dimensionality based merely on the type of recording that we're doing and the type of stimuli we're showing. All right. And so how do we get these large-scale recordings? So also feel free to ask questions in the chat. I can see the chat. So yeah, I'm happy to take questions whenever. All right, so we get these large-scale recordings using 2-photon calcium imaging with a microscope that can move quickly among many planes. We record at a rate of 3 hertz, capturing, in these older recordings, we were capturing around 10,000 neurons at 3 hertz. Now at Janelia, we're really lucky to work with some great microscopists here, and we're up to around 50,000 to 60,000 neurons at 3 hertz. But these are kind of our older recordings that were 10,000 neurons. And so there's a lot of activity you can see coming. So this is the image that's being shown to the mice at the same time as the neural activity. You can see there's activity all the time in the brain, but then when these images come in, there's a sharp increase in the amount of activity. And the circuit that we're studying, just for people who aren't familiar with mouse visual cortex, there are around 1 million neurons in mouse visual cortex. So it's an interesting circuit to look at. And so we take these recordings and we basically segment every single neuron in these recordings. We make these little masks, these different little colored discs. We take the sum of the pixels of every frame of the video in each of these disks, and that gives us one of these traces here across time. And then we can take these traces and take the average response of the neurons to each stimulus that we showed. We showed each stimulus for around, I think, 750 milliseconds, so a couple frames worth that we average. And then we basically end up with a matrix that's going to be 10,000 neurons by 2,800 stimuli. And then we asked this question, is neural activity low dimensional or high dimensional? And we found, in fact, it was actually neither of these things. It wasn't a conventional low dimensional code or a conventional high dimensional code. We found that the variance, the eigenspectrum of the data decayed like a power law of 1 over n to the alpha, where alpha was close to 1. And so we see this does suggest there's lots of dimensions of high dimensional activity, but it's not just this kind of flat eigenspectrum where these high dimensions have a lot of variance. There's kinds of these lower dimensions which have more variance, which kind of capture what we think are kind of the smoother, like the slower varying properties of the stimuli that the mice are seeing, that will drive more neurons in the population. And then you have these higher modes which drive smaller subsets of neurons and relate to smaller subsets of stimuli that don't overpower the fact that you can potentially still represent things smoothly across the population. And so in this paper, we propose kind of that neural codes are balancing capacity. So you want to encode as much information as possible. And to do that, you want to have as high dimensional of a code as possible. But at the same time, you want to keep kind of the smoothness principle that similar stimuli are represented similarly, so that every time I move a little bit, for instance, in this one-dimensional space, a completely different set of neurons doesn't start lighting up. If I move smoothly in stimulus space, hopefully it's just a smooth change of the neurons which are active. And so this kind of this power law is sort of this sweet spot that kind of can balance these two things. All right. Don't see any questions. So we're kind of going to use this structure. This power law structure is not just present in our recordings. We actually see it we see it also in our recordings of spontaneous activity. So this is the mouse is in complete darkness, and we record from visual cortex. We also see a power law decay, although it decays a bit faster. So there's less power in the higher frequencies, suggesting it's a little lower dimensional, but it's still a power law. So there's still many of these high dimensions represented. And then zebrafish whole brain activity, we also looked at the power law eigenspectrum, or we also looked at the eigenspectrum and found a power law. If you look at natural images, they also have this power law decay. And you can see this nice kind of representation of these kinds of principles of these branches, which are bigger, that are representing these lower powers. And then at the higher powers, you have actually similar representations, but at smaller scales. And then for the English language, we took the correlation matrix across words, and we got a power law decay as well. And then for social networks, we also got a power law decay for one of those Facebook data sets. And so we're going to kind of use these principles to try to guide us in creating a visualization algorithm for neural data, and hopefully, and I think I will say too, like hopefully our visualization algorithm is helpful for people modeling their neural data. That first you want to get a sense of what's going on in the data, and then you can try to build models of it. All right, so we have this large scale data, many thousands of neurons by many time points. And then, so there's a question. The power law exponent is the mean value of many trials data. Is the range of the power law exponent narrow among many trials? Yes, so we found, so we estimate the power law for stimulus-driven activity on 2 repeats of the same images. And so we have, maybe by trials you mean many like time points or many stimuli. And in that case, we do see that the power law kind of converges to 1 as a function of the number of stimuli. Anton asks, can you give an example of data that clearly do not obey such a power law? Yeah, actually, I'd have to look at this data more closely, but there's recent data from the Moser lab from grid cells, for many hundreds of grid cells. And if you look at that data, there's actually very clearly like 8 principal components that are very big. I think it's some number that you need to make the grid formation, and that data clearly doesn't have a power law. but maybe that's not the kind of data you're thinking of. But there are subsets of neurals of the brain that are probably not following a power law. So these things were limited. So this was cortical activity, and then for the zebrafish data, this was whole brain activity. And so we took a look at this is our spontaneous activity, and we're kind of looking at this data and wondering, what do we do with this? And so what we tried to do was create an embedding algorithm that could kind of help us visualize this data better, where we're going to put neurons that are correlated to each other close to each other, so that they're correlated in this time series, in this high dimensional space with many time points, and we put them close to each other in this one-dimensional embedding. And this gives us a plot that looks like this, and now you can see a lot more structure in the data. And so this is visual cortex, and this is the mouse's running speed. And for people who know mouse visual cortex, the neurons are driven by the mouse's running in spontaneous activity and evoked activity. And so you see these neurons that are clearly driven by running speed. There's kinds of variations in maybe their delay when running speed starts, and there's lots of different patterns that you can see come out when you resort this data. And so how do we get this visualization? So we have a few. I'm going to tell you what we found worked the best. We've tried a lot of different versions of Rastermap, but I'll tell you the version that works. So first thing we do is we reduce the data to a manageable size. So we take, in this case, we use k-means clustering, which kind of gives us like a coverage of the whole space, the whole neural space, and we get like 100 to 150 clusters from 50,000 neurons. And then we take these clusters and we rearrange them by similarity. And this is here where we apply this power law relationship. So we have this matrix of cluster correlations. So we have each of these clusters has a time series. And so we take these time series and we make their correlation matrix. And now we're going to want to rearrange these clusters so that correlated clusters are next to each other. And additionally, we're not just going to penalize if I was to penalize just clusters should be close to each other that are correlated, that would be like creating an off diagonal that was really positive. But I'm not just going to try to make an off diagonal that's positive. I also want to keep some of the global structure. So that would be keeping the local structure. So actually we try to match this correlation matrix to a correlation matrix that has a power law decay of eigenvalues, because this is going to have this slow decay off the diagonal with heavy tails. And so that will help us to try to preserve more of our global structure. But this is actually a parameter in the model you can change. You can try to make it more or less local or global, sort of like if you've run t-SNE before, like the perplexity parameter. And so we try to do this sorting. It's a version of sort of the traveling salesman problem, where you want to put things in the best order to optimize kind of that this correlation matrix is as similar to this one as possible. And so we use strategies that people use for traveling salesmen, which is taking subsets of nodes and greedily seeing where they best fit. And so we'll maybe take, for instance, this subset of nodes and see where it best fits, and we'll move it to whichever configuration has the highest score. And I can go into that in more detail if people have questions afterwards. But then basically that will give us a sorting that looks like this now. And you can see that now we've put things that are correlated to each other, close to each other, like even the off diagonal is pretty red, but also kind of even further off the diagonal, there's positively correlated clusters, because we've kind of preserved more of these global distances too. Okay, and so we only had 100 to 150 clusters, and that's partly because the traveling salesman algorithm doesn't really scale so well to more clusters. So we're working kind of on scaling that up a bit, and we have some other tricks for that I won't go into in this talk. But basically from there, we then upsample our clusters. So then we upsample by a factor of 10. So we'll have 1,500 nodes, and we'll upsample using linear interpolation between each of these clusters, because our clusters are in a row, so we can linearly interpolate between them. And so then we assign every neuron to a position along this one dimension, which is now has 1,500 different points. We put the neuron at the point in which it's most correlated with the time series at that given point. And then finally, we resort the neurons to create what we call super neurons. So we've put every neuron in the position that it's most correlated to, and then we put it in a plot, and you wouldn't be able to see anything actually unless I average across this dimension. So what we do is we bin neurons, and you can choose the binning of maybe like 50 neurons. And so that gives us kind of this plot where we can clearly see this structure better. So now we kind of say we're averaging over neurons rather than averaging over time, so that we can really capture single trial information about the brain. Because in the past, like each single neuron is noisy, but if I average over 50 neurons, which are correlated to each other, I'll get kind of a better representation of the data. Hopefully that's a less noisy representation of the data, and I can look at things on the single trial level that maybe I wouldn't have been able to in the past because of single neuron noise. And so that's the final picture we sort of get out. All right, does that sort of make sense? All right, so maybe I don't know how interested people are in this. There's been some recent debates on Twitter about how useful manifold embedding algorithms are. Personally, we care about preserving like local and global neighbors, and we use a metric that's slightly different from the paper. We can talk about it afterwards if people are interested. But basically what we care about is that if I have my original space maybe is 3-dimensional like this, I have maybe 3 neurons, and I have 3 time points, and these are all the different neurons and how they fire according to these 3 dimensions. And I want to basically make an embedding based on this 3-dimensional space. I want to put them in 1 dimension. And so what you probably want to do is you want to put all the blue points next to each other. They're all next to each other in the original space. You want to put all the red points next to each other. You want to put all the green points next to each other and all the yellow points next to each other. And then we can see that an embedding like this preserves kind of the local neighbors. And this is the benchmark that lots of people use for these embedding algorithms is how many of my original neighbors, so my neighbors from the original space, are still my neighbors in my embedding space. And so basically I can take my neighbors in my original space and ask what fraction of them are my neighbors in my embedding space. And that will give us, we can do that at different scales. And so at a scale of 10 of my neighbors, I've preserved 15% of my original neighbors are still my neighbors. And that's the case for Rastermap. It's a little slightly lower for TSNE. We tried several different perplexities for TSNE. We also tried the version of TSNE with 2 perplexities that you optimize twice. And that also didn't do better than Rastermap. And then we have also used UMAP compared to UMAP and PCA. And Rastermap did the best at preserving these. Basically, your local neighbors were similar to the neighbors you had in your original space. So this is that local preservation that works well. And then if you zoom out, I'll go through this quickly. Basically, you care about preserving local and global neighbors. In this case, actually, this wasn't a great fit for preserving global structure. You can see these green points probably should be close to these blue points. And so you would create an embedding that did a better job of preserving global distances would look like this. And so basically that you're looking at a score after you zoom out. And so what we found was that there's always this trade-off between local and global preservation. And what we found was that Rastermap still kept a high intermediate score and global score, so as well or better than TSNE, while giving us a better local score and preserving more of this kind of what we called kind of this high dimensional structure of the neurons. All right, and we can make raster maps where we sort images and neurons. This is actually shown in our paper, so I'm going to just kind of skip over this because it's not as exciting. We've also done Rastermap. We've also shown another thing you can do is take this matrix that you have that's neurons by time. You can predict it using the facial movements of the mouse, and this is stuff that my student Attica has been working on in my group. And she basically resorts. She takes the raster map and actually, instead of predicting single neurons, she predicts these super neurons in the raster map, these bin 50 neurons, and then she creates these plots down here that look like this. And so you have this lower dimensional space you can kind of work with that's less noisy, that you can create better predictive models and maybe get a better sense of how the neural space is representing behavior. Another way to use Rastermap is on behavioral data. So this is data that Lin Zong has collected in Marius Pakitsariu's lab. He's a postdoc there. And he has this task where mice run through 2 virtual corridors. And so there's a blue one and a red one. In real life, they're rocks, rock textures and leaf textures. And then one of these corridors, this red one, is rewarded at a random time. And basically the mice learn that they're going to be rewarded in this corridor, and you can see that they stop and lick in this corridor for these rewards, but they learn that in the blue corridor, they're not going to be rewarded, and they just run straight through, basically, and they lick much less often. And so you can see actually already that there's this activity in this is in visual cortex and a little bit, and surrounding areas, a little bit of PPC and other higher order visual areas. You can see this kind of sequence of activity through this corridor. And it's not perfect, but actually, we actually add this penalty. When I was talking about making Rastermap more local. We can add this penalty to the diagonal that helps a little bit, but then we have another trick that we've tried for temporal data, which is we take correlation matrices of these clusters. So we have these cluster time series. We can take these correlation matrices not just at a time lag of 0, but we can offset these traces and take their correlations at various offsets. And we can take the max correlation across a few offsets, and that will give us, so basically if I'm offset by this cluster by 3 time points, I'll still consider that I'll be correlated with it at a time lag of 3. And so that will show up in my time lagged correlation matrix if I take a max over at least 3 time points. And so if we do this sort of procedure, we get sequences that look much better like this, where we get these full sequences that you can see. And you can also see licking activity and other types of activity and potentially other signals that the brain is using potentially to learn this task we're thinking. And also, because we've made this one-dimensional picture, you can go through and look at what area these different neurons are in that are recorded here. This is the back of the brain, so this is more visual cortex. And when we're in the sequences, more of the activity is actually in primary visual areas. And then up here, these non-sequence related activities, like here, some of them are back there, but are more often kind of around visual cortex and not directly in visual cortex. So then you can take this data and sort of hopefully make some hypotheses about what you think these different neurons are doing, and you can look at these things across time and across learning. Okay, I am doing pretty good on time, I think. Okay, so you can also do this with Zebrafish data. This is an example of data recorded from Misha Aron's lab, where they showed a battery of different stimuli to the zebrafish, and Rastermap is putting neurons close to each other that have similar stimulus correlations and also similar motor patterns. So these are motor-driven neurons down here. And we can make these plots too. Again, we have this one-dimensional embedding, so we can see where these neurons are in the zebrafish brain that we're going through. We can also do this with not just neural data, but with voxels from Widefield imaging data. So we took this data set that the Churchland lab shared, which has Widefield imaging data during an auditory stimulus task. And so they have tones on the right and the left, and the mouse has to choose which tone, which has more tones, I believe. And they also have these kind of right and left visual stimuli during the task, but I think they're always, they're not using those as part of the behavior. But what's nice about those is then you can see during this right visual stimulus, there's a strong activation here in the purple, and that's where we've segmented things in the Widefield imaging as well. And so this might be a way for you to group voxels that might be, and visualize them that might be easier to see than other types of methods. Another thing is you can run these, you can run a model like this or an embedding algorithm like this on activations from artificial neural networks. So here's a QRDQN that's playing Space Invaders. So it has a value network and also a convolutional neural network that's processing the environment. And so here, it's playing the game, and this is what the activity looks like during the game. And so you see something neat here is you see this large value signal here that decays kind of as the agent kind of destroys the whole screen, and then it's going to start the next screen. And then the value increases again. And you can see these other sequences corresponding likely, because this is a convolutional neural network, to different patterns on the screen that are moving in correlated ways. But also maybe more high-level signals as well. There's a question from, I think I'll finish and then answer the question. Okay. Yeah. Unless you want to, I have one more slide actually after this.
Yeah, let's go ahead.
Thanks. I can skip over this. There's more sequences in this game, seaquest. But yeah, it's just another artificial neural network that you can look at. And finally, we have a GUI as well that you can use. So all of this code is on GitHub in the dev branch of Rastermap. And Atticus developed a new GUI for Rastermap, which makes it easy to look at these different patterns. Maybe just make it play faster while I'm here. Okay, and so in summary, we found Rastermap captures local structure better than t-SNE and UMAP. We have some extensions of the core algorithm for finding sequences and dynamics. There's also one for exploring fine-scale structure discovery. I didn't show that in my talk today. And we also have a GUI to explore the data. All right, so thanks everyone for your time, and thanks so much for inviting me to this workshop.
Sure.